Hi, good morning to all, and thank you for giving me opportunity to present my poster with the title Abundance of Sphingomyelin N-acyl-fatty acid chain length inversely correlate with the pancreatic cancer. So, why sphingolipid? Sphingolipids are the building blocks of the cellular and subcellular membrane and maintain the topology for the integral membrane protein. Sphingolipid known to manipulate a role in cell differentiation and the morphogenesis, and in principle it works with the oncogenic transformation and metastasis formation. To note, sphingolipid consists of sphingosine base, which is depicted in orange in here, uh, and fatty acid chain length, which is depicted in green in here. And the synthesis of this sphingolipid take place in endoplasmic reticulum with the incorporation of fatty acid chain length into the sphingosine base. What we did is within our cohort of the metapac study, what we did is we measure the metabolome analysis from the plasma EDTA plasma sample with the untargeted approach, and we measure global metabolite profiling as well as the lipid profiling. When we analyze this sample, and this is my part of result one, when we analyze the sample, we found a peculiar pattern with the sphingolipid metabolites, especially sphingomyelin and ceramides. And what we observed that the sphingolipid metabolites showed inverse correlation to that of their chain length. So as the chain length increases, the abundance inside the tissue decreases. To note, this is only substantial with the fatty acid which are derived from the fatty acid elongases, so evolves, but not with fatty acid synthesized by fatty acid synthase. So this is very peculiar for our understanding. When we check this or check this with ceramide, what we observe is ceramide also showed the same pattern, but in the more distinct than that of sphingomyelins. Yeah. When we compare this result with around 4,000 patients from the different cancer moieties, what we found out that there's a significant correlation of the chain length with PDAC, and some of the cancer do show significant correlation, but not as drastic as in PDAC. Also, when we check this with the chronic pancreatitis, we saw similar strong correlation. This hints towards not a correlation of the systemic inflammation, which we consider should be similar in chronic pancreatitis and PDAC. So, to conclude our part, sphingolipid abundance in PDAC is irreversibly associated with fatty acid chain length. This hypophenomena is not associated with systemic inflammation, and some of the cancer shows a similar trend. Thank you for listening to me.